Hello, good morning everyone. Myself Narinder Kaur, bio lecturer, Government Senior Secondary School, Jamsher Khas Boys. Today my topic is Human Genome Project. Human Genome Project is the most ambitious and the exciting scientific undertaking by the human beings. Human Genome Project is administered by the National Institute of Health and Department of Energy in USA, work started on this project in 1990 with the determination to map and sequence the complete set of chromosomes in 13 years. It is not a small job and labor and cost wise frustrating too. Study of this project will be undertaken under three steps: mapping, sequencing and the functional analysis. ਲਓ ਹੁਣ ਅਸੀਂ ਅੱਗੇ ਕਰਦੇ ਹਾਂ ਕਿ ਬਾਈ ਇਟ ਇਜ਼ ਕਾਲਡ ਏ ਮੈਗਾ ਪ੍ਰੋਜੈਕਟ. Human Genome Project is called as a mega project because human genome have 3 into 10 raised to power 9 base pair. The cost sequencing required is US 3 per $3 per BP. That is estimated cost in the beginning. The total estimated cost of the project would be approximately 9 billion US dollar. If it is obtained this sequence and stored in the typed form of books, then ਜੇ ਇੱਕ ਬੁੱਕ ਦੇ ਵਿੱਚ ਅਸੀਂ ਇੱਕ ਪੇਜ ਦੇ ਉੱਤੇ 1000 ਲੈਟਰ ਲਿਖੀਏ ਔਰ 1000 ਪੇजेस ਦੀ ਇੱਕ ਬੁੱਕ ਬਣਾਈਏ, then 3300 such books would be required to store the information of DNA sequencing from a single human cell. To store this data, the computational device will be required which was named as the bioinformatics. ਅੱਗੇ ਅਸੀਂ ਸਟੱਡੀ ਕਰਾਂਗੇ the goals of the HGP. It is to study the sequence of all the base pairs in human genome and to identify and determine the function of all the genes and to store the information obtained from DNA sequencing in database and to identify and point out the specific genes that cause the genetic disorder and to transfer the technologies to other sectors like industries and the agriculture and to add the possible ethical, legal and the social issues that may arise from the project. Then salient features of the Human Genome Project. The Human Genome Project has 3.1647 billion base pairs. The average gene has 3000 bases, but their sizes vary greatly. The larger gene contains 2.4 million bases. The total number of genes present in the human genomes is 30,000. There are maximum number of genes present in the chromosome number 1, that is 2968, and the minimum number of genes, that is 231 in the Y chromosome. Almost 99.9% of nucleotide bases, they are exactly same in all the human beings, and there is a difference with only 0.1 nucleotide base that difference are the function of more than 50% discovered gene, they are not yet known. The related sequences of the nucleotides make up a major portion of the human genome and only less than 2% that are codes for the proteins. About 14 million locations have been identified where single base DNA difference occur in human genome. This information is going to help to find the chromosomal location for diseases associated sequences and tracing human history. ਹੁਣ ਅਸੀਂ ਅੱਗੇ ਕਰਾਂਗੇ what are the applications of HGP. It is the basic understanding of the human genetics, then reveal the genes responsible for the diseases like cancer and diabetes and provide information that will prevent inherited diseases and lead to treatment of genetic disorders through the gene therapy and help plan genetically modified diet. ਅੱਗੇ ਹੈ ਫੰਡਿੰਗ ਕਿ ਇਸ ਇੰਨੇ ਬਿਗ ਪ੍ਰੋਜੈਕਟ ਨੂੰ ਕਰਨ ਦੇ ਲਈ ਫੰਡ ਕਿੱਥੋਂ ਆਇਆ। ਸਭ ਤੋਂ ਪਹਿਲਾਂ US ਡਿਪਾਰਟਮੈਂਟ ਆਫ ਐਨਰਜੀ, ਦੈਨ ਨੈਸ਼ਨਲ ਇੰਸਟੀਚਿਊਟ ਆਫ ਹੈਲਥ, ਦੈਨ ਵੈਲਕਮ ਟਰਸਟ UK, ਐਂਡ ਦਿਨ ਹੋਰ ਕੰਟਰੀਜ਼ ਨੇ ਸਪੋਰਟ ਕੀਤਾ ਲਾਈਕ ਫਰਾਂਸ, ਜਾਪਾਨ, ਚਾਈਨਾ ਐਂਡ ਜਰਮਨੀ। ਦੈਨ ਮੈਥਡੋਲੋਜੀ ਕਿ ਇਹਦੇ ਲਈ ਕਿਹੜੇ ਕਿਹੜੇ ਮੈਥਡ ਜਿਹੜੇ ਆ ਇਨਵੋਲਵ ਹੋਏ। ਇਹ 'ਚ ਦੋ ਮੇਨ ਅਪਰੋਚਿਸ ਆ। ਇੱਕ ਹੈ ਟੂ ਸਿਕੁਐਂਸਿੰਗ ਓਨਲੀ ਦਾ ਕੋਡਿੰਗ ਪੋਰਸ਼ਨ, ਜਿਸ ਨੂੰ ਐਕਸਪ੍ਰੈਸ ਸੀਕੁਐਂਸ ਟੈਗਸ ਦਾ ਨਾਮ ਦਿੱਤਾ ਗਿਆ। ਦੂਸਰਾ ਹੈ ਟੂ ਸਿਕੁਐਂਸਿੰਗ ਦਾ ਕੰਪਲੀਟ DNA, ਜਿਸ ਨੂੰ ਸੀਕੁਐਂਸ ਐਨੋਟੇਸ਼ਨ ਜਿਹੜਾ ਆ ਇਹ ਕੀ ਆ ਕਿ ਦਾ ਟੋਟਲ DNA ਫਰੋਮ ਏ ਸੈੱਲ ਲਿਆ ਗਿਆ, ਫਿਰ ਉਸ ਨੂੰ ਆਈਸੋਲੇਟ ਕੀਤਾ ਗਿਆ, ਦੈਨ ਕਨਵਰਟਡ ਇਨਟੂ ਦਾ ਰੈਂਡਮ ਫਰੈਗਮੈਂਟਸ of relatively smaller size and a very long polymers, and there are कई technical limitation in sequencing very long pieces of DNA and cloned in a suitable host using vectors. ਵੈਕਟਰ ਜਿਹੜੇ ਕਿਹੜੇ ਯੂਜ਼ ਕੀਤੇ ਗਏ ਇਹਦੇ ਲਈ BAC ਐਂਡ ਦਾ YAC, ਔਰ ਹੋਸਟ ਜਿਹੜਾ ਯੂਜ਼ ਕੀਤਾ ਗਿਆ ਦੈਟ ਵਾਰ ਦਾ ਬੈਕਟੀਰੀਆ ਐਂਡ ਦਾ ਯੀਸਟ। ਦੀ ਸਿਕੁਐਂਸ the segments were sequenced using automated DNA sequence that worked on the principle of a method developed by the Frederick Sanger. Sanger ਨੂੰ ਮੈਥਡ ਫॉर ਡਿਟਰਮੀਨੇਸ਼ਨ ਆਫ ਅਮਾਈਨੋ ਐਸਿਡ ਸੀਕੁਐਂਸ ਇਨ ਪ੍ਰੋਟੀਨ ਦੇ ਵਿੱਚ ਵੀ ਕ੍ਰੈਡਿਟ ਮਿਲਿਆ ਸੀਗਾ। ਦੀ ਸਿਕੁਐਂਸਿੰਗ ਵਰ ਦੈਨ ਅਰੇਂਜਡ ਬੇਸਡ ਔਨ ਸਮ ਓਵਰਲੈਪਿੰਗ ਰੀਜਨ present in them, then specialized computer based programs were developed. These sequencing were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed in May 2006. Some repetitive DNA sequence known as the microsatellite shall be explained in the DNA fingerprinting. Thank you.
ਹੈਲੋ, ਹਾਂਜੀ, ਮੇਰਾ ਨੈਕਸਟ ਟੌਪਿਕ ਹੈ ਜੀ DNA ਫਿੰਗਰਪ੍ਰਿੰਟਿੰਗ। ਇਸ ਵਿੱਚ ਕੀ ਆ ਕਿ ਅਸੀਂ ਇਹ ਕਹਾਂਗੇ ਪਰ ਸਭ ਤੋਂ ਪਹਿਲਾਂ ਕਿ ਵਾਟ ਇਜ਼ DNA ਫਿੰਗਰਪ੍ਰਿੰਟਿੰਗ। DNA ਫਿੰਗਰਪ੍ਰਿੰਟਿੰਗ ਨੂੰ DNA ਪ੍ਰੋਫਾਈਲਿੰਗ, DNA ਟੈਸਟਿੰਗ ਔਰ DNA ਟਾਈਪਿੰਗ ਦਾ ਨਾਮ ਵੀ ਦਿੱਤਾ ਗਿਆ ਆ। ਇਟ ਇਜ਼ ਅ ਫੋਰੈਂਸਿਕ ਟੈਕਨੀਕ used to identify individuals by characteristics of their DNA. A DNA fingerprint is a small set of DNA variation that is more likely to be different in all the unrelated individual, thereby being unique to individual as are fingerprints. ਜੇ ਅਸੀਂ ਅੱਗੇ ਇਸ ਦੀ ਡਿਸਕਵਰੀ ਦੀ ਗੱਲ ਕਰਦੇ ਹਾਂ, it was discovered by Sir Eli Jeffrey 1985. India ਦੇ ਵਿੱਚ in India it was started by Dr. B.K. Kashyap Lal ji. Sir Eli Jeffrey is called the father of DNA fingerprinting, whereas Dr. Lalji Singh is called the father of Indian DNA fingerprinting. DNA ਫਿੰਗਰਪ੍ਰਿੰਟਿੰਗ ਜਿਹੜੀ ਆ ਇਹਦੇ ਲਈ ਅਸੀਂ ਸੈਟੇਲਾਈਟ DNA ਜਿਹੜਾ ਆ ਯੂਜ਼ ਕਰਦੇ ਆ। ਇਹਦੇ ਲਈ ਅਸੀਂ ਪੂਰਾ DNA ਯੂਜ਼ ਜਿਹੜਾ ਉਹ ਨਹੀਂ ਕਰਦੇ। ਅੱਗੇ ਹੈ ਕਿ ਇਹ ਕਿਸ ਪ੍ਰਿੰਸੀਪਲ ਤੇ ਡਿਪੈਂਡ ਹੈ। It has been found that the DNA of an individual carries some specific sequence of nucleotides which do not carry any information for proteins. The specific sequence of nucleotide, they are repeated many times and for many places throughout the length of DNA. These sequences are very specific in each individual. These sequences are called as VNTRs. VNTRs means variable number tandem repeats. During the DNA fingerprinting, the sequence of the nucleotide of the VNTR is studied. Process of DNA fingerprinting. DNA molecules, they are separated from the sample. Sample ਅਸੀਂ ਕੀ ਲੈ ਸਕਦੇ ਹਾਂ? Blood, serum and other appropriated fluid from the personal items like toothbrush, razors, hairs, etc. Then the DNA molecules are then cleaved into small pieces with the help of restriction enzymes. These DNA fragments contain VNTRs. Then DNA fragments are then stored out according to their length on an agarose gel slab by the electrophoresis technique. These fragments get arranged along the cell according to their length. Then double stranded DNA is now split into the single strands by placing the gel into an alkaline solution. Alkaline solution means it's a solution of NaOH. Then these isolated fragments in the gel, they are copied onto a nylon or nitrocellulose membrane sheet placed on the gel. Pressure is applied evenly to the gel to ensure the good and even contact between the gel and the membranes. The membrane is then exposed to hybridization probes. Special DNA probes are prepared in the lab. These contain the sequences of the nucleotide complementary to those which are on the VNTRs. These probes are made radioactive. The radioactive DNA probes bind to the repeat sequence on nylon sheet. इसनों कहने हैं, this is कहने हैं hybridization. This is called the hybridization. Then an X-ray film is then exposed to nylon sheet to mark the places where radioactive have been bonds to the DNA fragments. These places are marked as dark band when X-ray film is developed. This process is called the radiography. The dark band on X-ray film repeat the DNA fingerprints. Identify casualties or persons missing in action. The DNA fingerprinting methods will be far superior to dental records. Blood typing strategies. Okay, forensic or criminal use. FBI and police labs around the world have begun to use DNA fingerprints to link suspects to the biological evidences. ਉਹ ਕਿੱਥੋਂ ਲੈਂਦੇ ਆ DNA from the blood, semen or hair. ਅਗਰ ਉਹਨਾਂ ਨੂੰ ਥੋੜਾ ਜਿਹਾ ਪਾਰਟ ਵੀ DNA ਦਾ ਮਿਲ ਜਾਂਦਾ ਆ ਤਾਂ ਉਸਨੂੰ ਉਹ PCR ਟੈਕਨੀਕ ਨਾਲ ਉਸਦੀਆਂ ਬਹੁਤ ਸਾਰੀਆਂ ਕਾਪੀਜ਼ ਬਣਾ ਲੈਂਦੇ ਆ ਤੇ ਫਿਰ ਉਸਨੂੰ ਯੂਜ਼ ਕਰ ਲੈਂਦੇ ਆ. Another important use of DNA fingerprint. Thank you.